Biology Part 22. DNA coding strand at the beginning of the gene and becomes attached to the DNA at this point. This seems to break the hydrogen bonds linkage, the two strands of DNA, so that the double helix onward in the gene region. The DNA-dependent RNA polymerase molecule now travels along the gene and new molecules. Having brought them into the organic structure, we do get to our translation. In the next stage of protein synthesis, the coded instructions in a molecule of mRNA must be translated into a sequence of amino acids forming protein chain. This process requires a molecule which is attached to a ribosome. Well, the ribosomes are small cell organ cells about 20 centimeter in diameter. Each they are 20 nm, that means 20 nanometer. They are 20 nanometer in diameter, each containing about 50% of the ribosomal RNA and rRNA and about 50% of the protein. They consist of two particles of different sizes called 40S and 60S subunits. These names reflect the relative masses of the particles in the Seeberg units. See index and glossary. Both kinds of subunits are assembled in the nucleus. The two subunits of the ribosome remain separate, and the molecules of mRNA are available. Then they lock it on the special sequence of nucleotides at one of the mRNA called ribosome binding sites. Within the ribosomes there are tRNA sites where a triplet code word or codon or mRNA can become attached to the base pairing of the molecule. And there are each tRNA, there are moleculotides, anticodon, and precisely this purpose is there. So the tRNA is linked to the specific amino acid, and of course the position that they provide over there. When two acids are given there, they accordingly make their own way and find the solution to it. Next to codon into position, as this process continues, the ribosome travels along mRNA strand, adding more amino acids to the growing proteins chain. Accurate interpretation of mRNA message is only possible because, without exception, each tRNA molecule has a particular anticodon specific for only one amino acid. Consequently, a given mRNA codon is always translated in the same way. The meaning of the 64 different mRNA codons appears to be the same of the same index that we have been wearing. So when I don't wear, only you get to it and put an explanation to it. This corresponds to amino acid methionine and is called initiation codon. The methionine therefore occurs in the beginning of every protein chain, although into many cases it is removed by enzyme action before the protein is folded into this final shape. We can certainly have the different understanding to it. At the end of the transcription, the polymerase and enzymes recognize the stop sequence, which becomes the detached gene region away from the DNA molecule, and suddenly has a double helix configuration.